Now we can move on to the mechanism behind the cytoplasmic male sterility. First of all, we can observe the cytoplasm. In those sorts of plants, they having two kinds of cytoplasm. The first one is the normal one, and the second one is an aberrant one. Aberrant means abnormal one. These cytoplasmic male sterility, they may be controlled by some of the extranuclear genetic materials. That means they may be mitochondria, chloroplast, or some other cytoplasmic factors. That's why they shows non-Mendelian inheritance.
We have seen that the maternal inheritance, that is mainly because of some of the cytoplasmic factors or cytoplasmic genetic materials. Here in the case of CMS, that is also controlled by some of the extranuclear genetic materials or cytoplasmic genetic materials. But apart from the other maternal inheritance, here some of the genes those are present in the nucleus, they are also comes in role. For example, RF. RF is the restoration of fertility genes. They are actually located in the nucleus, not in the cytoplasm. The real function of it is that it can restore the fertility. That means, see, a cell that's having an aberrant cytoplasm or aberrant cytoplasmic genetic materials, but it's having a normal RF gene in the nucleus. Then this RF gene that can restore the sterility of that plant. You know that these nuclear genes or RF genes, they are inherited in accordance with the Mendelian inheritance. So if there are two copies of the functional RF, then definitely it can restore the sterility. If the RF gene that is heterozygous in condition, then also it can restore the sterility. If the RF genes are homozygous recessive one, then it cannot restore the sterility. That means the cytoplasmic male sterility that can be observed in a plant that having a defective cytoplasmic genetic material, as well as also having homozygous RF recessive alleles. Now we will get more details in this schematic representation.
In this picture, there are four different lines of plants are there: A, B, C, and D. Now we are going to cross A with B and C with D. Here in this picture, A and C lines are considered as the female plants because we are going to pollinate the flowers of A and C with the pollen from B and D lines. Hence, the B and D lines are considered as the male plants. We can definitely reverse the conditions. That is, if you are going to pollinate the flowers of a B with the pollen from the A, then the female plant will become the B and the male plant will become A. But here in this picture, we are considering A and C as the female plants and B and D are the male plants. Coming to the genetics of these plant lines, both of them are recessive RF alleles, it's having a normal cytoplasm. And coming to the D line, it's having normal cytoplasm and the RF genes are homozygous dominant. You all know that the RF genes are located in the nucleus. These RF genes are located in the nucleus. And coming to the A line, it's having a sterile cytoplasm, but the nuclear genes or alleles, both of them are recessive RF. So they are having a sterile cytoplasm, and for the RF allele, it shows a recessive homozygous condition. And this is the case with both the A and the C lines. So this B line is having normal cytoplasm, but both nuclear RF alleles are recessive, and for the A line, it's having sterile cytoplasm, but both of its RF alleles are homozygous, are recessive, and this is also the same with the C plant line. And about the D line, it's having normal cytoplasm, but both of its restorer genes are dominant one. I have already told you that both B and D lines are male lines because the pollen is actually derived from these B and D lines. See, this is the schematic representation of a pollen. It's having a little bit cytoplasmic content in it. See this green area that shows the cytoplasmic region, and this is a normal nuclear region that is having RF genes. And coming to the female plant, that is A and C lines, the egg is actually derived from these female lines. Egg actually bears an enormous cytoplasmic content in it. In addition to that, the RF genes are also present in the nuclear region. Now what we are going to do is that we are going to cross the B line with the A. So the resultant progeny, all of them are having sterile cytoplasm, and both of its RF genes are homozygous recessive one, because see we are going to cross it with this egg. So this cytoplasmic content actually inherited from the female plant only. So all of the progeny will have a sterile cytoplasmic constituent. And about the nuclear genes, they are actually inherited in a Mendelian manner. So RF is crossing with RF, again this RF with this RF, then RF with the RF, the small RF with the small RF. So all of the progeny having both of its allele are homozygous recessive nature. Now we are going to cross the C line with the D line. C line is the female one. Its genetic constituent is that it's having a sterile cytoplasm, and both of its nuclear RF genes are homozygous recessive. So egg is having this genetic constituency. It's having an enormous cytoplasm, as we have said before, and the pollen is actually derived from the D line. So it's having the genetic constitution as the normal cytoplasm, and both of its nuclear genes, RF genes, are homozygous dominant in nature. But we need to only consider about the nuclear gene because the pollen having a little bit cytoplasmic content in that. So the cytoplasmic genetic materials are inherited from the mother plant only. So in the progeny, it's having a genetic constitution of homozygous RF genes. That means it's having both dominant and recessive RF genes in the nucleus, and for the cytoplasmic content, it's a sterile one because the mother plant itself is a sterile one, and the cytoplasmic content is actually inheriting from the mother plant. So all of the progeny having the sterile cytoplasm. And about the nuclear genes, see the RF is crossing with the RF, and again this RF is again RF, and this small RF with the capital RF, then this small RF with the capital RF. So that means all of the progeny having a heterozygous RF gene, and its cytoplasmic content is the sterile one. So after the cross with the B and the A line and the C and the D line, we have obtained two kinds of progenies. One is having sterile cytoplasm, and both of its RF genes are homozygous in nature, and other plant having Its cytoplasm is again a sterile one, but its nuclear genes RF are heterozygous in nature. So this is a male sterile plant. Now what we are going to do is that we are going to cross these two lines. That means we are going to cross this line with this line. For that we are considering the C line can be denominated as C. So this C line as the female one, not the male one, because it's the male sterile one, and this is the male one. So the pollen is obtained from the D line. This can be denominated as the D line. So the pollen is getting from the D line, and that pollen is used to pollinate the flowers of the C line. This is the constitution of the egg. It's having enormous cytoplasm. This is sterile, and both of its nuclear genes are homozygous recessive. And about the pollen, it's having heterozygous RF alleles, but it doesn't have any cytoplasmic regions because it's pollen. Normally, its cytoplasmic content or genetic constitution is sterile, but we don't need to concern much about the cytoplasmic constitution because this is the pollen. And now what we are going to do is that we are going to pollinate this egg with the pollen. All of its progeny having sterile cytoplasm because the cytoplasm content is inherited from the female plant. And about the nuclear gene, see RF is crossing with RF. So one progeny is heterozygous, and again RF, small RF with the small RF. So the remaining half is homozygous recessive. See again this small RF with the capital RF again forms a heterozygous progeny, and then a small RF with a small RF. That means half of the progeny having homozygous recessive genetic constitution, and the remaining half is having heterozygous genetic constitution for that RF gene. But all of its cytoplasmic content is sterile one. See, all of its cytoplasmic content are sterile one, and half of its genetic constitution is RF, and the remaining half having RF and RF. So this is a male sterile plant, and this is a male fertile plant. That means in this way we will get half of the seeds are fertile and the remaining half of the seeds are sterile. That's how we can restore the male sterile plants in the population. That's why the D line is known as the restorer line, and this is the maintainer line. The maintainer line may be having both of its RF genes are homozygous recessive, but its cytoplasmic constitution may be sterile or normal. That's how these lines can maintain male sterile plants in the population.